Let's begin by going over exactly what we'll cover in this video. First, we'll examine the historical origins of medicines and how traditional remedies influenced modern pharmaceutical development. Next, we'll investigate initial laboratory evaluation methods for potential new treatments. Then we'll look at how promising compounds are assessed in controlled human studies. Finally, we'll consider the importance of scientific verification before medications receive approval. Throughout history, the natural world has provided many therapeutic substances. Plant species synthesize various chemical compounds as defense mechanisms against insects and microorganisms. Remarkably, some of these natural substances can effectively treat human ailments. For centuries, people observed which plants seemed to offer medicinal benefits. These observations formed the foundation of traditional healing practices worldwide, many of which eventually led to contemporary pharmaceutical products. Consider aspirin, widely used for pain relief and fever reduction. This common medication originated from compounds identified in willow bark. Long before modern pharmaceutical science, people recognized willow's pain relieving properties. Similarly, the cardiac medication digitalis was developed from compounds present in the foxglove plant, which had historically been used for heart ailments. Microorganisms have also yielded important medicinal compounds. A noteworthy example comes from Alexander Fleming's laboratory work. While examining bacterial cultures, Fleming observed a curious phenomenon. A mold contamination had created a bacteria free zone in one of his culture dishes. This mold, identified as penicillium, was producing a substance that eliminated the surrounding bacteria. Fleming had discovered penicillin, inaugurating the antibiotic era in medicine. In contemporary times, pharmaceutical manufacturing occurs on an industrial scale. Although many medications still derive from natural substances, chemists frequently modify these compounds to enhance effectiveness and minimize unwanted effects. Before any medication becomes available to patients, it undergoes extensive evaluation through multiple testing phases. This rigorous assessment process ensures both safety and efficacy. Laboratory evaluation represents the first phase of this process. During this stage, researchers examine how compounds interact with human cells and tissues in controlled laboratory conditions. These experiments provide preliminary insights into potential therapeutic mechanisms. However, laboratory studies using isolated cells cannot fully predict how substances will behave in complete biological systems. For example, assessing cardiovascular medications requires evaluation in organisms with functional circulatory systems, not just isolated heart cells. Following successful laboratory studies, researchers conduct animal studies as part of preclinical evaluation. These investigations serve multiple purposes. They assess therapeutic effectiveness, identify potential toxicity issues, and help determine appropriate dosage ranges. UK legislation mandates testing proposed medications on two different mammalian species before human trials begin. This requirement generates significant debate. Some advocate for animal testing as a necessary safety measure, while others question its ethics and seek alternative methods. Compounds that successfully complete preclinical evaluation advance to human clinical trials, which proceed through several distinct phases. Initially, researchers administer experimental medications to healthy individuals. These early trials focus primarily on safety assessment rather than therapeutic benefit. Researchers begin with minimal concentrations of the compound, gradually increasing amounts if participants tolerate the substance well. When safety data from healthy individuals appears favorable, researchers then evaluate the medication in patients with the targeted condition. These studies determine optimal therapeutic concentrations, amounts that maximize beneficial effects while minimizing adverse reactions. To accurately assess effectiveness, researchers randomly assign participants to different treatment groups. Some receive the experimental medication, while others receive a placebo, an inactive substance appearing identical to the medication under investigation. This methodology allows researchers to distinguish actual medication effects from perceived improvements that occur simply because someone believes they're receiving treatment, the placebo effect. Many clinical studies employ blinding techniques to minimize bias. In blind studies, participants remain unaware whether they've received the actual medication or placebo. Double blind studies extend this principle further. Neither participants nor the immediate research team knows who received which substance until the study concludes. This approach prevents unconscious biases from influencing how researchers evaluate participants or how participants report their experiences. Before publication, research findings undergo peer evaluation. During this process, independent scientists scrutinize the methodology and conclusions to verify scientific validity and procedural integrity. This critical review mechanism helps prevent unsubstantiated claims about new medications and ensures that only thoroughly validated information reaches medical practitioners and patients. Developing new medications involves extensive resources, often requiring many years of research and significant financial investment. However, this comprehensive approach helps ensure that medications reaching patients offer genuine therapeutic benefits with acceptable safety profiles.